Hi all, Dr. Clark here again, General Biology lecture. Today we're going to lecture on proteins. Okay, the macromolecule proteins, polymer proteins, biomolecule protein, okay, just the entity of proteins. This picture here is a picture of a capsid, okay, and a capsid is a protein coat around nucleic acids in viruses, and this is the 3D rendition of the Zika virus. Okay, some of you might remember the Zika virus scare, okay, during the Olympics in Brazil, and causes birth defects. But this is the 3D rendition of what the protein coating around the nucleic acids looks like. Okay, and so proteins are more than just food. I mean, we often think of it as being food, but proteins are building blocks of really everything. It's the way you look is due to your proteins. Okay, your ability to break down food, your ability to have an immune system, your ability, to really do anything, have homeostasis, is really the result of proteins. Now, it's not exclusively proteins, but nonetheless, proteins have a major role in all those activities. Okay, so we're going to look at proteins just in general. Okay, so proteins are macromolecules or biomolecules that are made up of amino acids. Okay, and this will play a very important role as we move further into the course. We'll talk about the link between nucleic acids, so DNA, and amino acids and proteins. Okay, because it's DNA that codes for your proteins. So DNA codes for what amino acid is going to be laid down to make this giant polymer called a protein. Okay. Now, when you lay down an amino acid and you chain some of them together, the link between them, the bond between them, is called a peptide bond. Okay. And if you have a whole bunch of them, it's called a polypeptide. Okay, so often another term for proteins are called polypeptides, because this is the bond that makes up these long polymer chains of amino acids and results in a polypeptide or protein. Okay, and you can see how this is formed by this diagram here. You can see that we have an amino acid here that ends in a hydroxide, an amino acid here that ends in a hydrogen. Okay. We know that this is a dehydration reaction because water is formed. It's not showing you, but energy is supplied here to form this peptide bond between carbon and nitrogen and to form this polypeptide chain, amino acid linked to another amino acid. Okay. Now, interestingly, if you look at the ends of these other amino acids, okay, there's a hydroxide group, there's a hydrogen group, so it's, as you can imagine, it's easy to link another amino acid over here and another one over here, and most of our proteins that we know of are lots of amino acids linked together. Okay, hundreds of amino acids linked together to form simple proteins. Okay, sometimes 300 to even 1,000 amino acids might be linked together to form these long polypeptide chains. So again, amino acids is the small molecule or the simple structure. Okay, but even on top of that, amino acids are made of other smaller structures, and that is that they're made of an amino group, a carboxyl group, and then a functional group. Now, all amino acids are made of amino groups and carboxyl groups. Okay, and what determines what amino acid you're dealing with, whether that's valine or lysine or you know, whatever amino acid there is. Okay, there's 20 of them, and we'll talk about them in later lectures. But the functional group, or what we call the R group, is what determines the structure, the function, what it can bind to, what kind of properties does it have, is it polar, is it non-polar, okay, and what kind of shape is the molecule going to fold into, and we'll get to this in a second. Okay, so it's that functional group that's really important for the actual identity of the amino acid. Okay? And like I just said, there are 20 different types of amino acids. Now, there's a little bit of caveat to that. There are 20 different types of amino acids that occur naturally in nature. Okay? Chemists have the capability and have designed amino acids. Okay? So chemists have made their own amino acids. I think they've made 4 amino acids, 4 different types of amino acids. So in the grand scheme of things, there's 24 amino acids, but only 20 of them occur naturally. Okay? The other interesting piece about amino acids is that when a meteorite or an asteroid hits our planet, and we've done this before, if you swab that rock, or that substance, okay, if you swab it, you can pull off amino acids. So it means that there are amino acids not just on our planet, but there's also amino acids, the building blocks to proteins, elsewhere. Does that mean that there's life elsewhere? We're not going to discuss that because there's really no evidence for it. Okay, but this does supply a little bit of evidence that at least it's possible. Okay. Protein structures can be very complex. Like I said before, you can have hundreds of amino acids linked together. Okay? But because there are only 20 amino acids, it's the order of the amino acids that determines kind of the structure of the protein. Now, when I say there's only 20 amino acids, I mean there's 20 amino acids for all organisms. Okay? A plant has the same 20 amino acids. A bacteria that might be on a pen has the same 20 amino acids. The fungus that's growing, in your fridge because you haven't cleaned it out, same 20 amino acids. You, same 20 amino acids. Okay? But what makes us not resemble a bacteria or the plant or the fungus is the arrangement of the amino acids, which is dictated by our DNA. Okay? And we'll get to this. Okay? The other thing that's interesting is even though there's only 20 amino acids, there are hundreds, probably 150,000 I think described so far, maybe a few less, maybe a few more, proteins on this planet. Okay? So we know that there's about 150,000 different proteins. Okay? So it's not just the order of the 20 amino acids, but it's the order of the 20 amino acids and then how those 20 amino acids can fold and twist together. Okay? And we'll get to this in a little bit. That will determine what the protein looks like, and that would then determine what the structure looks like. Okay? So polypeptides, these long amino acid chains, will fold together, okay, and fold over on each other to determine the protein's function or determine the protein's shape. Okay? And we know enough now that we know that a lot of proteins, after the initial folding process, they'll also combine with other proteins to make a much larger protein. Okay? And one of the reasons that we know this is because we know that there's only roughly 25,000 genes in the human genome. Okay, so we have 25,000 different codes for proteins. But we know that there's over 100,000 different proteins that humans construct. Okay? And so, based on that, you would know that either the genes are coding for more than one protein, which is quite possible, okay? Or the genes are coding for proteins, but the proteins are combining with each other, which is also possible. And now we know that both is true. So there are 4 general levels of protein structure: primary, secondary, tertiary, and quaternary. Okay? Now the primary structure is just the chain of amino acids. Okay? And this is the base level. Guys, this is kind of the background, the core level. If you alter a genetic code, you alter the amino acid on that chain, which changes your primary code, which changes secondary, tertiary, quaternary codes. Okay? Or structures, sorry. Secondary structure is how that primary structure folds. So is it twisting? Is it forming a coil? Is it forming a helix? Is it forming a sheet and folded over on top of each other? Okay. How does the protein itself initially fold? Tertiary structures are more 3D shaped. So you might have one region of your amino acid chain that goes into a helical shape, another region that goes into a sheet shape, like a beta sheet, okay? And then maybe you have some kind of coil also, or multiple beta sheets, multiple coils, multiple helixes, okay? That's a tertiary structure. And quaternary structure is taking a few of these tertiary structures and sticking them together, making a much more complex protein. Okay? Now, a lot of this work was done by this man, okay? Linus Pauling. Okay? And I think you probably heard me talk about him before. Okay? Linus Pauling is the only individual so far to win 2 Nobel Prizes by himself. Okay? So now there are 4 people that have won multiple Nobel Prizes: Barton, Curie, Pauling, and Sanger. Okay? But Linus Pauling is the only one to win 2 of them by himself, and besides Madame Curie, Marie Curie, he's the only person to win Nobel Prizes in different fields. So Madame Curie won it in physics and chemistry, and Linus Pauling won it in chemistry and in peace. Okay? So these 2 books are just excellent books. Linus Pauling is a very unique character, very unique background. Okay, he was born in Portland, Oregon, so he's an American scientist. He dropped out of high school because, according to him, it wasn't challenging enough for him, and then entered college at Oregon State University in Corvallis, Oregon. Got his bachelor's degree, and then he went on to get his PhD in California. Okay, and just a well-established chemist. Lots of work. He's credited with not only figuring out how proteins fold, okay, but also providing a lot of the evidence and a lot of the research needed to figure out that DNA was a double helix. Okay, so Linus Pauling won the Peace Prize in 1954 for the Nobel Prize in Chemistry in 1954 for the discovering of protein folding, amino acid folding, okay, chains, and then won the Peace Prize in 1962 for speaking out against war and hate crimes and things like that. Okay, I suggest both these books. I'll put a link in below. Check it out, or one of these books. Doesn't really matter. Okay, so proteins. So let's go through those structures again. If it's a primary structure, it's an amino acid sequence, okay, and what we call a polypeptide chain. Okay. Now, again, like I said, this is determined by the DNA of the organism, okay, and any change in the DNA, at least any change in the gene region of the DNA, will result in a different primary structure. Okay. So this determines all the other levels of protein structure. The secondary structure is when a polypeptide chain folds over on itself. Okay? So again, I said that some amino acids are nonpolar, some amino acids are polar. Okay? And so you're going to have regions that are nonpolar that you typically are going to be on the inside of the protein, protected from water. Okay? And then you have regions that are polar that will form hydrogen bonds. Okay? And by having these structures, you can get these unique, nice shapes like coils or helixes or sheets. Okay? And that's really the secondary structure to it. The tertiary structure is often the final form of certain types of proteins, and they'll be in 3D shape, and that's really just how they twist and how they fold, okay, and all the possible combinations along that polypeptide chain of helixes and sheets and coils, etc., folded together in one big form. Okay? So when we talk about proteins, whether we're talking about enzymes or proteins that are building tissue structures or encapsulating DNA, whatever it is that we're talking about, they'll either be in the tertiary structure form or the quaternary structure form. Okay? And this really just is determined by, well, what's the protein's function? Okay? How large is the protein? Okay? So tertiary is kind of one genetic code. Okay? Can be very long, okay? But one genetic code determining that structure. Quaternary structure is typically multiple genetic codes. Okay? So you get multiple polypeptide chains being linked together to form one. Okay? So here you can see what I'm talking about. Here's that primary structure dictated by your DNA. That primary structure is going to fold or twist in a certain way and form hydrogen bonds between the polar regions. Okay? And then a tertiary structure, you might have these beta sheets, which are shown here. You might have these alpha helixes. Okay? You might have some coils here. You might have all kinds of things within a tertiary structure. Okay? And then you can get these tertiary structures combining together to form a quaternary structure. Okay? So just like what we were talking about at the very beginning of this lecture series, we said structure determines function. It's the exact same thing with proteins. Okay? We're not just talking about structure of like thumbs and fingers and body function. Okay? We're talking about the structure at the macromolecule level determines its function. Okay? So if there's an opening or an activation site and something can fit in there, and this is a protein that is considered an enzyme, okay, it's specific to that one function. Okay? And we'll come back and talk about these. Okay? Now, there are some components in nature that can change the protein's shape, that can change the protein's how it's folded, okay, by breaking hydrogen bonds. Okay? And when that happens, it's called denaturization or denatured. Okay? That means the protein is in a non-natural state now. Okay? And the hydrogen bonds that were holding those coils and those sheets together and those helixes together are now being broken, and the protein's unfolding. Okay. There are a couple ways at which this can occur. You can have temperature changes that can do this. Okay. Mainly increases in temperature, which will break those bonds, increase the amount of energy, okay, that's needed to break the simple hydrogen bonds. Remember, these are hydrogen bonds, so they're fairly weak forces, okay, when they're in isolation. So when there's not tons of them, it's a fairly weak force. It can be broken very easily, okay, and temperature can do that. Okay. pH can also do this. Okay. Okay? And so certain proteins have optimal pHs. They also have optimal temperature ranges. Okay? And outside of that optimal pH or temperature range, you can start getting denaturization, or the protein be denatured. Hydrogen bonds broke, okay, and the protein is misfolded. Okay? When this occurs, the protein becomes inactive. Okay? And so, meaning that the protein will no longer work. And the other thing is, until recent technology, this was a permanent process. Okay? The denaturization of a protein is permanent. Okay? Once the bonds are broken and new bonds form, and the protein changes its shape and it no longer has any function. Okay? That being said, there has been research done to reverse the denaturization process. Okay? And if you're truly interested in this, you can shoot me a line. Okay, and I can give you some papers that you can read up on it, on reversing denature, and you can check it out. It's in the preliminary processes right now, and there's been some pretty good research that's been done on it, and it's important, and we'll talk about why a little bit later.
This familiar gelatin dessert actually is a good example of coagulation of proteins into a three-dimensional latticework that entraps water molecules to produce a semi-solid gel. This animation explains the physical chemistry behind the process. Proteins are synthesized by polymerizing amino acids. The polymerization occurs by repeatedly forming peptide bonds that link individual amino acids together into a chain. There are three structural features that influence the three-dimensional shape of a water-soluble protein. Primary structure is the peptide bond between individual amino acids that creates a long chain of connected amino acids. These long chains of polymerized amino acids have hydrophobic, water-repelling, and hydrophilic, water-attracting projections that are oriented perpendicular to the chain, as shown in this illustration of a growing protein chain. Secondary structure is the helix that the protein chain curls into as a result of hydrogen bonds and other weak forces. Tertiary structure is created when the protein molecules fold back on themselves outside of the helical segments to put the hydrophobic portions to the interior and the hydrophilic portions to the exterior. Several helical regions can exist in different portions of the molecule. When the protein has folded and refolded to reach its most stable configuration, it will have mostly hydrophilic amino acid residues on the exterior and mostly hydrophobic residues directed into the interior. When natural proteins are subjected to physical or chemical treatment, their structure changes and they become unnative or unnatural. We call that process denaturation. In this example, heating the proteins in solution imparts energy to the protein molecules. This added energy is enough to break the relatively weak forces that hold the protein in its refolded and helical tertiary and secondary configurations. As the process of denaturation proceeds, the protein molecule unfolds more and more, and the internally directed hydrophobic regions now become exposed on the outside of the molecule. The peptide bonds are largely hydrophilic. Once these segments are set free from each other, they attract water molecules. The recruitment of water molecules entraps the water molecules in close proximity to the protein strands. The hydrophobic portions of the molecules are also exposed. This situation is unfavored because the hydrophobic portions of molecules are not stable in an aqueous environment. Hence, upon unfolding, the hydrophobic regions on individual protein molecules will associate with hydrophobic regions on other protein molecules. This situation encourages the association of these protein molecules into larger and larger random three-dimensional structures. the molecules aggregate into very large water-insoluble collections that are quite randomly assembled. As the proteins denature, latticework structures grow amorphously and attract the solvent water molecules into cell-like structures. The self-associated water molecules, in groups, adhere to the surface of hydrophilic regions of the protein, while hydrophobic regions of the protein dissolve into each other and provide the energy to retain the structure. As this process continues irreversibly, all of the protein molecules are recruited to this large insoluble mass in a randomly organized structural framework that contains entrapped water molecules. One example of the consequences of unfolding and reassociating protein molecules is coagulation of egg white. Frying an egg is no more complicated than denaturing the egg white protein. The assembly of irreversibly denatured protein molecules results in formation of a solid gel. The gel entraps water molecules inside the white into a semi-solid structure, which holds its shape under normal conditions. Other examples of denatured protein assembling into three-dimensional structures include the baking of yeast-risen bread, coagulation of meat proteins by cooking in such products as hot dogs, and in the solidification of gelatin upon cooling of a solution. Denaturation and coagulation of proteins is a complex, irreversible process, but the study of denaturation has allowed us to better understand the three-dimensional structure of native proteins.
Okay, so you can see there that the video says that it's a permanent, irreversible process. Okay, sort of. Okay, like I said before, there are researchers that have reversed the denaturation process. Now, the protein that they reversed it on didn't come back exactly like it was before the denaturation process, but it's still functional. Okay, and so we'll keep in tune to this, and if I find more information about it, I'll share it with you. I haven't dove into the literature in a while. A couple years ago, they were able to reverse the denaturation process. This is important if you think about, from a medical standpoint, if you think about individuals who have extremely high fevers, by being subjected to a high fever, 105, whatever, for a long period of time, the denaturization process can start to take place. Okay, the protein can start, the hydrogen bonds can start to break, unravel, and then those proteins are toast, and normal functionality within that organism's body is gone. Okay? And that's why a lot of people would die from having high fevers, because the proteins that are needed for the biochemical pathways to dissolve food, or to run neuron processes and these kind of things, they're destroyed. Okay, so we'll come back to that at a later date. Okay, so that kind of brings us to a specialized type of protein, which we call enzymes. Okay, so enzymes are either in that tertiary or quaternary form, okay, and they have an active site. We'll come back to enzymes and talk a lot more about them, but we're going to just briefly hit on them so you know a little bit about enzymes. They have this activation site, this open site that has a precise fit with some other molecule. Okay? When that other molecule fits in that activation site, the protein changes shape. Now, it doesn't change shape by flexing its muscles or anything like that, because we're talking about macromolecules. It changes shape because of the charge, okay, of whatever that chemical is that just encountered that molecule. So if it's positive, then and the charge on the protein is negative, they might be attracted and pull towards each other a little bit. Okay, we call this an induced fit. Okay, and so it's very specific to one substrate, one enzyme. Okay? Now, when this occurs, that substrate comes in, encounters the activation site of the enzyme induced fits around it. Okay, then that process can allow for bonds to be separated or bonds to be formed, okay, and it can catalyze a reaction. Most of you have heard of a catalyst of a reaction will speed up a reaction. This is true, it does speed up the reaction, but the reason why the reaction moves faster, okay, is because the amount of energy needed for that reaction to occur, either the separation of substrates into two substrates or the formation of two substrates into a product or whatever, it needs energy. In both cases, it needs energy for that to occur. Catalysts or enzymes, okay, that decrease the amount of energy. We call that activation energy. It decreases the amount of energy needed for that to occur. By doing so, the reaction is sped up. And in all cases, or in some cases, okay, the reaction would never even occur without that protein, without that enzyme. Okay? So it's not a matter of just speeding things up. Sometimes those enzymes are needed for the reaction to occur at all. Proteins fold specifically, we already talked about this, so you're going with that amino acid chain, folding into secondary structure, folding into a tertiary structure. Okay. Now often we ask ourselves, well, how do proteins know what way to fold, or what's the specific folding process? Okay. Now this is kind of, I want to say, kind of a black box of biology right now, and there's lots of researchers that are interested in this piece, okay, because we know that there are special proteins called chaperone proteins that will take a protein that's misfolded and fold it correctly, and will do it over and over and over again. Okay? How they fold it correctly, we don't know, okay? And that's really a very important piece to a lot of research that could be done on things like cystic fibrosis, which is a lung disorder that's caused by two recessive alleles, okay, and it's caused by the misfolding or defective chaperone proteins. Alzheimer's, which is also linked to these defective chaperone proteins. If we can understand how chaperone proteins work, specifically how they work, then maybe we can start looking at how to correct the defects that we see in things like cystic fibrosis and Alzheimer's. Okay? So we know this much about chaperone proteins. We know that if you have a misfolded protein, okay, that it'll encounter a chaperone protein. It has the capability of encountering chaperone protein. It moves inside these proteins. A cap is placed over the top. The protein, at least as much as we know, is isolated inside a chamber kind of thing. It's refolded. We don't know this piece, like how is it refolded, okay? But we know that it comes out with the correct folded protein, okay? And this happens over and over and over again, okay? So there are lots of researchers out there, many major universities that are spending, massive amounts of time and large sums of money to figure out this process, this chaperone protein work. Okay. All right, so we'll stop there. That's just a general overview of proteins. Next we'll talk about nucleic acids and lipids, okay, and probably some carbohydrate stuff also. Okay, see you next time.